Good afternoon and welcome to the Iowa Learning Farms 2026 Conservation Webinar Series. I'm your host this afternoon, Liz Ripley, the Conservation and Cover Crop Outreach Specialist. And we're thrilled today to have with us Clarisse Siababa, a graduate research assistant here at Iowa State University, discussing how they're working to harness local woodland resources to enhance bioreactor microbiome for water quality improvement. If you're new to our series, welcome and thank you for joining us. If you are a longtime follower, thank you again. If you have questions for Clarisse, feel free to enter those into the chat and we will get to the Q&A portion of today's webinar following her presentation. So with that, Clarisse, the virtual floor is all yours.
Okay, thank you so much, Liz, for that introduction. So, wait. So again, we are very thrilled today to be invited to share the project that we've been working on for the past year. The Forestry on Top project is actually a USDA NIFA AFRI-funded project headed by Dr. Michelle Supir from the Department of Agricultural and Biosystems Engineering. And this project is made possible by a lot of efforts from our collaborators and partner institutions. So we're very grateful for the invitation from the Iowa Learning Farms. And again, thank you everyone for joining us, taking the time off and listen to the project that we're working on. So please be guided by the outline of my presentation. I'll be giving a brief background of some terms that are important for this presentation. Some of you might already be familiar with this, or some might be new to the terms. And then I'm going to discuss the research gaps and the rationale behind doing the project and focusing more on the project objectives, the research questions that we are trying to answer. And then we're going to discuss the current laboratory works, the methodologies involved in the research work, and focusing more on the preliminary results. That's what we want to share to everyone here on three important aspects. The wood extractives, these are actually the wood components or compounds that have been extracted from the trees, the wood metabolites, which are products of the wood metabolism, and the microbial denitrification aspect. This is more on the nitrate removal of the different tree species. And then we're going to wrap everything up with some future directions and key takeaway from the presentation. So let's get started. So if you came to Iowa 200 years ago, you would have seen that it is highly dominated by prairie, significant wetland, and woodland system. But today you'll see that it is actually a highly agricultural production system with over 95% of the ecosystem that have been developed or transferred to agriculture. And now, Iowa is mainly composed of agricultural cropland. Based on the National Land Cover Database, more than half of Iowa's total land area is used for cropland. So in this map, Iowa is designated by the red star, with over 85% of the crop area or cropland used for corn and soybean production. And based on the Soil Survey Geographical Database, Iowa is composed of poor or very poor soil drainage. It is shown by the blue to dark blue color in this map. And again, this is Iowa as depicted by the star. So what actually is the implication of having poorly drained or very poorly drained soil compared to to a drained soil? If your farmland is undrained, just like this figure on your left, undrained soil actually traps water. It prevents the volume change and prevent water from escaping, leading to increased pore water volume. As a consequence, the crop actually have stunted growth because the root system cannot reach into deeper parts of the soil. So meanwhile, if you have a drained land or a drained soil, just like the figure on the right side, the drained soil allows water to escape, lowering the water table. So we have lower free water level, allowing crops to grow better. It can lead to extensive root network for better crop production. And Iowa has by far the most tile drain acres in the country. So this is from the USDA Census of Agriculture. So one of the solutions that or conservation practices use for the poor drain soil is the use of agricultural tile drain. So in this map, areas with blue or dark blue regions are heavily tile drain. And again, Iowa is the most by far tile drain area in the country. So tile drainage has been used for a couple of years now for increasing the agricultural production for areas with poor drained soil. But aside from that, even though it is very efficient for enabling agricultural production in areas with poorly drained system, it has also increased nitrate and other pollutant in the water because of the accelerated movement of water. And with that, it contributes to high nutrient loading and river discharge in the Gulf, which can lead to hypoxic zones. So this map shows you areas in the Gulf that are with evident low dissolved oxygen concentration, leading to hypoxic areas or the dead zone. And when we have depleted areas of oxygen, it is unsuitable for marine and aquatic life. So it is kind of harmful for our aquatic ecosystem. And Iowa alone contributes to 11 to 52% of the nitrate loading to the Mississippi River Basin, as shown in this map, because there are a lot of tributary rivers contributing to the efflux of the nutrient to the Mississippi River Basin. So with this, what should we do? We should look for conservation practices to mitigate the impacts of high nutrient loading and river discharge. And one important edge of field approach is the use of wood chip bioreactors. So wood chip bioreactors have been used to remove nitrate from agricultural tile drainage before it enters the waterways. So what is actually a wood chip bioreactor? So in agricultural farmland, you excavate a chunk of land and or soil and you put in wood chips. So the water, the agricultural tile water from your field, which has high nitrate concentration, gets diverted to this wood chip system before it enters the waterways. And the important drivers in this wood chip bioreactor are the microorganisms. So the microorganisms here, we have different microorganisms. They convert nitrate from your tile drainage water to a less harmful form. The final product is usually the dinitrogen gas, giving us cleaner water in our waterways. And the process involved here is what we call the nitrification. This is just the nitrate removal process governed by our microorganisms. And the microorganisms in our system here in the wood chip system use the wood chips as their food, as their carbon source. So the wood chips from different trees can act as the food source for our microorganisms. So with that knowledge, we can actually enhance nitrate reduction by looking into the different wood chips that can be used as food source for our microorganisms. And in the Midwest region, particularly in Iowa, there is an abundant supply of food species that are underutilized but can potentially be used as substrates for our wood chip bioreactors. And instead of just throwing them away or burning them, using them as firewoods, we can actually use this wood species for other economic viable outcomes or outlets. And we're trying to look into the use of low historically marketable wood chips for their potential application as our substrate. So some examples of wood species with low historic marketability includes hackberry, cottonwood, and mulberry, compared to those with higher marketability such as walnut and oaks. But again, we should study more on what is present on our wood species. So there is still a gap between the effect of the wood species and their extractive. So again, the term extractive, this is just the wood compounds that can be found on our wood species. So we want to know, is there really an effect on the individual wood species and the wood compounds and how they will influence the nitrate removal ability of our wood chip bioreactor? How would it affect the microorganism present in our bioreactor? So the rationale behind this project, we think that if you choose the right wood, it might actually increase the amount of nitrogen in the form of nitrate that is removed by our bioreactors. And this can be done by better understanding the wood properties. So once we know the wood properties, how the wood extractives would impact the microorganism, we can better optimize our wood chip bioreactors. We want to bridge in the gap by identifying the wood species, the extractives or the compounds that from the wood, and this can lead to a more optimized denitrification or nitrate removal in our wood chip bioreactors. Now, our main research question is we want to know the relationship between the wood species, the extractives, and the microbiome, and how they will influence the nitrate removal for an improved water quality outcome. And to answer this overarching research question, we have three sub-questions. First, what is really on the wood? How can we know if the wood species are different from each other? So for the first question, we're going to characterize the wood species in terms of the wood compounds isolated from them, the microorganisms that can be found in this wood species, and their nitrate removal efficiencies as individual wood species. The second question is, can we actually improve water quality by choosing the wood species or the right wood species? So in here, we have done small batch scale reactor test for the individual wood species. And we want to know if there is really difference in how they remove nitrate from our initial nitrate concentration. After we have identified the wood species with the highest or fastest removal efficiencies and the lowest removal efficiencies, we want to create a wood chip blend that would be used for our larger scale setup. So for the second question, we're using the small scale setup, and we want to further use it for a more larger scale setup using columns before we actually put them out in the field. So the overall goal is to develop an optimized wood chip blend that would enhance nitrate removal in wood chip bioreactors to have an improved water quality outcomes and also sustainable use of our locally available wood resources. So these are just some photos of our wood chip sampling. So we have collected 13 wood species with 3 biological replicates from our farmers and landowners. So thank you very much for your help. And we have chipped them up in our farm. And these wood species are actually different from each other in terms of their porosity or growth rings. Some wood species are characterized by having diffuse porous, wherein the pores in the ring are highly uniform to one another. Though that we also have wood species with ring porous classification, wherein the pores are variable from the early to the late wood stage. So we have smaller pores during the late wood stage. And the semi-ring porous, there is a gradual decrease in pore size for our pores for the rings. So some examples of wood species: for ring porous, we have oaks. For diffuse porous, we have the maples. The semi-ring porous, we have the walnuts. The figure on the right side is an example of a tree cookie showing you the distinct areas of the wood. The darker or the inner core is called the heartwood, and the outer core, the newer layer, is called the sapwood. And I'm going to briefly discuss more about this too in the succeeding slides. So again, we have 13 wood species. These are just some examples of the wood species that we're using. This is the complete list of the 13 hardwood species for the project. The one in the red text is the black walnut. This is the semi-ring porous category. The one in the blue text is the ring porous, and the one on the green text, these are the diffuse porous ring species. Now, looking into the wood composition. Sorry, taking a water break. So for the wood composition, so from the previous slide, I've shown you a tree cookie. So again, the inside or the inner core is known as the heartwood. This is where we can find the wood, more of the wood compounds and the extractives. The newer layer here is known as the sapwood. This is where the living parenchyma tissues can be found. where the food of the living tree are sourced out. And then looking deeper into the trunk of the tree, this is where we can find the hemicellulose, cellulose, and lignin. These are the or the bigger molecules present in our tree. And we want to further look into the extractives. This is a very important component of the wood because some extractives can actually inhibit the nitrate removal. And some of these extractives are the tannins, the ellagic acid, the gallic acids that I'm going to show you later. Now, for the methodology, so we have collected the wood chips from our landowners and farmers. We have air dried the wood chips and used them for cold water solubility extraction, wherein we extracted the metabolites from the woods. And we have used these extracts for both colorimetric assay and the metabolomic analysis using the liquid and gas chromatography analysis for both targeted and non-targeted identification of the compounds present in our woods. We also did the batch reactor experiment using small mason jars for identifying the nitrate removal efficiencies of the individual wood species. We all started with the same concentration of nitrate and look into how long it will take for our individual wood species to remove nitrate, either partially remove nitrate or completely remove nitrate. And we measured nitrate and nitride concentration for different sampling points. And we're currently working on the DNA extraction directly from the wood chips for us to identify the microbial community present on the individual wood species. Now, for the results. So again, I'm going to focus into three important aspects. The wood extractives, again, these are the wood compounds isolated from the different wood species, individual wood species. The metabolites, these are the products of metabolism from the wood. And the microbial denitrification, this is just the nitrate removal ability of our wood species. Okay, so for this slide, I want to show you that the different wood species have different metabolite composition. These are just 3 out of the 13 wood species, but I want to highlight that the number of metabolites isolated from wood species are very different from one another, ranging from 700 metabolites from honey locust down to just 170 metabolites from cottonwood. Overall, we have identified 1,851 known metabolites under 20 superclass categories. So this superclass category are the annotations or the functions of the wood species based on different databases. Out of this 1,800 identified wood metabolite, there are only just over 30 metabolites that are consistent on all 13 wood species. So meaning the different wood species are very unique from each other in terms of their metabolite composition. But interestingly, although they highly varied on the number of wood compounds that can be identified from the individual wood species, the top 3 that are dominant for or dominated all 13 wood species include the ones in the blue color. These are actually the lipids and the lipid-like molecules, such as the fatty acids that they use in order to grow, and also the ones in the purple and the pink ones. These classifications are the organic acids or the carbohydrates that are being used as the food source of our trees. Next, in terms of the total phenolics. So the phenolics are plant chemicals with antioxidant properties. And based on literature, they are known to inhibit microbial denitrification or the nitrate removal efficiency. And overall, the 13 wood species have varying total phenolic concentrations. So this graph shows you the 13 wood species classified based on their vessel distribution. The ones on the top are the diffuse porous tree species. The ones in the middle are the ring porous wood species. And we only have black walnut as our semi-ring porous species. Interestingly, we have identified the wood species highlighted in the red box as the wood species with higher or highest total phenolics compared to the other wood species. And we think, and also based on literature, this might actually produce lower nitrate removal efficiency. So once you have higher phenolics, there is a chance that the nitrate removal might be not as fast as the other wood species. And that's what we want to show in the succeeding slides. So this slide is for specific for total phenolics. The next slide is for the tannic acid. Tannic acid is an important extractive, and it is comprised of either gallic acid or ellagic acid. These are just examples of tannic acid. Out of the 13 wood species, we have 9 wood species with tannic acid content greater than 5 ppm. So having a tannic acid content greater than 5 ppm can be disturbing already in our drinking water, so we don't want this concentration. But for our wood species, we have black walnut with the highest tannic acid concentration. So it is something to look into. Having high total phenolic and high tannic acid content, what is the implication of this in the nitrate removal of the specific wood species? Now, this slide is the overview of the nitrate removal efficiencies of the 13 different wood species. The figure on the upper left, this is the cold water solubility extraction that we did in the lab for the different wood species. And the ones at the bottom, this figure is an example of the batch reactor using mason jar experiment for all 13 wood species with 3 biological replicates each. So let me walk you through this slide. So we have the different wood species on the x-axis. So we have the 13 wood species, and the y-axis is the nitrate concentration. So we started with an influent concentration of around 30 ppm or 30 milligram per liter of nitrate. We want a concentration that is as low as possible after a certain time of incubation. So for this graph, this is after 72 hours of incubation, and after that specific time, there are wood species that reached almost zero nitrate concentration. Again, that's what we want. The lower, the better. So from a 30 milligram starting milligram per liter concentration, we have 2 wood species with almost zero nitrate concentration, and that is your cottonwood and hickory. But again, they are from different types of porosity. We have different wood species that still has high concentration, such as black walnut here, and we have the oak here. So we want to take a look. Maybe this wood species, they need just more time in order for them to completely or partially remove nitrate. So we further extended the incubation time after 72 hours of incubation, and this is what we found. This might be a busy slide, but let me walk you through this slide. So we have the 13 wood species here. They are classified based on the different vessel types because we want to know if there is actually difference in the vessel distribution and its impact on the nitrate removal. So this slide will show us that there are wood species with higher or faster reduction and slower reduction. What we want is a complete decline early on the incubation time. So for hickory and cottonwood, there is a complete decline or almost complete decline starting at 60 hours of incubation. But it needs until 72 hours in order to completely remove the nitrate out of the small jars. After the hickory and cottonwood, the next one with fastest removal or faster removal are the hackberry, the black cherry, and the black maple wood species. And those with the slowest removal are highlighted in this side. We have the black walnut, red oak, and honey locust. These 3 wood species, particularly the black walnut, has the highest total phenolics and the high tannic acid concentration. And out of thousands of metabolites that we have identified, we want to specifically look into the metabolites or the wood compounds or extractives that are associated with the nitrification. And this graph shows you some of the metabolites that are positively or negatively associated with the nitrate removal efficiencies or performance rather. So we have the ones on the pink bars. These are those that are positively associated with nitrate removal efficiencies. Some examples are siloinositol, silobiose, glucose, erythritol. These are compounds that are known to be sugar alcohols or plant alcohols or carbohydrates in general, which can enhance microbial carbon utilization. The ones in the blue bars are the ones that are negatively associated with the performance, such as gallic acid, hydrofilacin B, and the eupatoroxin. These are metabolites that negatively associated with the nitrate performance because they are either antimicrobial agents or quorum sensing inhibitory compounds, which actually makes sense because if you inhibit the quorum sensing of the bacteria, or basically if you inhibit the bacteria to talk to one another, they cannot receive the signal from their neighboring bacteria if the quorum sensing is inhibited. So it might actually lead to less nitrate removal performance. And for the next steps of our project, we're going to, again, still working on the extraction, the DNA extraction from our wood chips. We want to analyze the microbial community present and create a wood chip blend. So when you say create a wood chip blend, we want to mix different wood species and see if it will actually lead to a better and faster nitrate reduction. So after our small scale setup, we're going to use our blended wood chips in our column bioreactors before using them for field demonstration. And again, the overall goal is to develop a wood chip blend that can be used out in the field. So some key takeaway for the presentation: even though we know that wood chips have been used widely for conservation practice, the performance can still vary substantially depending on the wood species that you're using. And the type of wood species can highly affect the nitrogen removal efficiency. From our preliminary results, out of the 13 wood species, we have identified 2 wood species with the fastest nitrate removal efficiency or nitrate removal rate within 72 hours. We have hickory and cottonwood, with hickory having around 99.89% reduction and cottonwood 99.83% reduction, almost the same. And black walnut also showed one of the slowest nitrate removal efficiencies. You need more time in order to reduce nitrate, you need more than 600 hours in order to reduce nitrate. And the key takeaway here is that the different wood species, they have different wood compounds. Again, these are the extractives. We're still looking into the specific wood extractives from the individual wood species and what is the implications of this wood metabolites on denitrification. But we want to share with you that different wood species, their extractives can impact nitrate removal and the microbial community in the wood chip bioreactor. And these are just some of my references. And I would like to thank our collaborators, the USDA NIFA Iowa Learning Farms, the Water Quality Research Lab, and the Germs Lab for their efforts in bringing together this project. And a lot of great hands helped us in making this project possible. So thank you, everyone. And I hope I shared some important knowledge to you, and I'm open for questions.
Excellent. Yeah, questions are coming in. So if you want to go ahead and go through our next few slides as those get entered in. Key partners of our webinar series, we want to give them a thank you. They are the Iowa Department of Agriculture and Land Stewardship, our Iowa Department of Natural Resources, the Iowa Nutrient Research Center, and the Leopold Center for Sustainable Agriculture. And as always, if you attended today's webinar for a CCA CEU, please be sure to email Elena Whitaker at alenaw@iastate.edu by 5 p.m. today with the following information noted on the screen. That includes your name, the name you used to watch the webinar if it's different than the name you have registered with the association, and then your CCA number that we can get this credit submitted on your behalf. And then our next slide here is a voluntary demographics survey. We appreciate everyone who has completed it so far for 2026. You only need to do it once. It's fairly short, should take you about 3 minutes or less. You can access that survey by the URL on the screen there, iowalearningfarms.org/survey, or simply scan that QR code. Like I said, it's very short, easy to do on your phone. If you want to give that QR code a scan, or again, check out our website, iowalearningfarms.org/survey. Okay, I think the next one is for next week's webinar. So that's on July 15th. We're talking on-farm energy opportunities, distributed wind, with Kathy Evanson McDermott and Sophie Farr. So that should be a great opportunity to learn more about that. And then our last slide is where you can find this recorded webinar and all previous webinars. That's either on our website at iowalearningfarms.org/webinars. We host those via Vimeo, or we've also started a YouTube channel where you can search these webinars both by year and by topic. So you can check those out. Now, there's been a lot of questions that have come in, and I did a little bit of research on one of these because there was a question about how many bioreactors are in use on how many acres in Iowa. So there's roughly about 300 bioreactors across the state. And as of 2024, the Iowa Nutrient Reduction Strategy had recorded about 11,000 acres treated. So there's been a few more installed in 2025, but that's kind of where we are on that. So you mentioned looking at a variety of species. And just to clarify, were pine or fir included in these? I know the species primarily were chosen because of their Iowa ability to grow, because trying to grow them more locally to where these are being installed. But can you remind us if any pine or fir
Yeah.
species were included?
For our project, we focus on hardwood, so we don't have pine or conifers. But I know another undergraduate student who used conifers for their potential use as bioreactors, and I think it's uploaded in one of the websites in Iowa State as well that they have successfully identified some red cedar to be used for bioreactors with around 80% reduction, if I'm not mistaken. But again, for this project, we focused on hardwoods. We didn't use any pines or conifers.
And so kind of related to this, so far in what you've studied or found, are there some species that are just not performing and we shouldn't be including, or is it currently still something is better than nothing?
Okay. Surprisingly, there are those that really stand out because they just need a shorter time to reduce nitrate. But out of the 13 wood species, they can actually still be used. It's just that you need more time for them to reduce nitrate because, as I've shown you in the graph, even though black walnut needs 672 hours, it can still reduce nitrate up to 94%, and it's still a good value. So we can still use those 13 wood species. It's just that some takes more time than the others.
So kind of maybe clarifying how bioreactors utilize and remove nitrate, because there's a question here about possibly recovering nitrate and how the land application of the wood are once saturated with nitrate. So maybe clarifying again kind of that denitrification process
Mhm.
and whether or not any nitrogen is retained within the wood chips.
Okay. So again, for the wood chip bioreactor, the system uses the wood chip as the food source of the microorganisms. So the microorganisms present in our system are the ones responsible for converting nitrate to a different form of nitrogen. So it can either be as nitrogen gas, which is what we wanted, but other forms of nitrogen can also be formed, especially if it's just partial nitrate removal. But the purpose of the bioreactor is to completely or partially remove nitrate. So we want nitrate to be completely removed out of the system. I'm not really sure if there are still nitrate or nitrite that are maintained in the system. Based on our small scale bioreactor setup, we have seen wood species that can generate nitrite along the way, but there are peaks in nitrite levels. But overall, this 13 wood species can remove nitrate, completely remove it to nitrogen, and that's what we want.
So just as a point, if you wanted to follow up on those conifer potentials in bioreactors, Elena did link the story about that
Yeah,
research in the
yeah,
chat, so you can
yeah.
explore that a little bit more. Um, so you mentioned, time is definitely a part of it, but as part of this project, is there a component analyzing the cost and availability of each of these wood species compared to its level of nitrate removal, or is that more down the line?
Um, sorry, can you repeat the question in terms of
Yeah, so the
nitrate?
question is, did you analyze the cost and availability of each of these wood
Oh, okay.
species compared to its nitrate
Hmm.
removal? And maybe we're just not
Okay.
quite ready to do that analysis, but something
Yeah.
down the line as we think about this
Yeah.
as well.
So for the project, it's not right now it's not part the cost analysis of that, but I'm sure that we have ideas on like the abundance of the different wood species available across Iowa. I can look more into that with some of our partners because we have used this wood species since they are highly abundant in the Midwest, and we can really source them out in this region. But again, for the project, I'm not familiar with looking into the cost benefit analysis of that, so I'm very sorry for that.
We're still trying to figure out how to kind of
But
maximize the
yeah,
mix and the species, and
yeah,
then we can run the numbers.
yep.
Um, So part of this project is looking at, timber management, and that was partially how these species were selected, was to figure out, okay, how can we take these lower value woods species
Hmm.
that are beneficial to remove for timber management. Uh, so, and you kind of already answered the second part, but that all the species did, in fact, remove nitrate just under different
Yeah,
time scales.
yeah, different time scales.
Would you say that there's any high efficiency species of wood that stood out to you?
Oh, right now there are two wood species that really stood out, and I was surprised that it only took them like 3 days. But take note, this is a small scale one. Out in the field, it might take them more time. We're not really sure about that, but we have the initial data that can show that hickory and cottonwood are so far the two wood species that really stand out for me. Hickory, based on what I've read, is more of a moderate marketability compared to cottonwood with really low marketability because it is soft and not really as durable as as walnut or oak. So with that, there are things to do with cottonwood instead of just throwing them out. But again, as I've said, this is just a small scale setup, the small jars. Well, we can give you more updates once we use the wood chips in a bigger scale, like the columns or out in the field, if they can still maintain that removing nitrate in just 3 days. That would be great, but it's too early to say how long it will take for them to completely remove nitrate. But those two wood species really stand out.
So there's a question here. So bioreactors typically, I think this is a good visual description. If you imagine a school bus, so
Mhm.
that's usually the length,
The
depth, and
length.
width of a standard bioreactor. So as we think about how many wood chips are needed to fill a space that could hold a school bus, is what we're looking for each of these. Um, so there's a couple questions. One of them being about, do you anticipate seeing a different microbiome, excuse me, present based on the location of where the trees were harvested or grown?
Right now, that's our next step to look into the microbiome aspect side of things. But based on the previous research that we've done, there's definitely differences in the microbiome composition depending on where you find them or source them out. But the thing is, if you identify the core microbiome responsible for the nitrification, the location is just a small aspect of that. So that's what we want to find out for our next step, since I'm doing the DNA extraction from the individual woods and further look into the microbial community. By then, I can better answer this question if there are really differences in the woods, in the microbiome collected from different locations, because there might be differences. We're not yet sure about that.
Do you know about how long the trees were uh, deceased before the wood chips were made? Were they wet or dried?
I'm pretty sure that we've collected some that have been cut out a few months or so before they were chipped. Some are actually fresh. That's why we dry them out before using them because there's still like moisture inside of them. So since we outsource them from different farms, some of them have been there in as logs for quite some time. Some are just freshly cut out, and I'm not sure if they're even those out in your backyard or neighbor's backyard that you can just use and you can donate it to us. I don't know. Just kidding. So we use it for our wood chips. But again, we chip all of them at the same time and we dry them out in the lab before using them because some of them are still moist.
Um, so do we have a good idea of what the species are currently in bioreactors? Is it a mix?
Right now, I'm pretty sure that it's a mixture of everything. With we were trying to reduce the percentage of using oaks. Um, um, there's certain amount or ratio of the different wood species that you can use. But in our lab, we're using a mixture of different wood species from our different collaborators. If I'm not mistaken, we're using wood species from Golden Valley and J. Petticourt, and it's just a mixture of different species.
So there's a couple questions here. I'm gonna kind of group them together. So the experiment results for faster nitrate removal from like hickory or cottonwood imply that those species might be used up quicker, requiring an early replacement of the media. Um, and then there was another question about like how often do they need to be replaced?
Okay. Out in the field, I'm pretty sure that a wood chip bioreactor can stay there for 10 to 15 years, depending on the woods that you use. So for hickory and cottonwood, even though they can remove nitrate faster compared to the others, it doesn't really necessarily mean that all of the nutrients are used up. It's just that there might be specific nutrients from those wood species that helps the microorganism to convert nitrate faster compared to others, and that's what we're trying to look at. If there are core metabolites that are only found in cottonwood or core metabolites that are only found in hickory, that makes them an ideal, quote unquote, we're not yet sure, ideal candidates for that. But again, if it's out in the field, it takes like 10 to 15 years before you can replace the wood chips in the bioreactor.
Um, so there's a question in here, and it's kind of maybe we'll get Billy on in a little bit, or Pooja, but we do have a project that's also looking at the size and makeup of the wood chips themselves. So while you're focused on the wood chip species, there's a
Yes.
kind of partner project that we're a part of as well, funded by the Iowa Nutrient Research Center, that's looking at the wood chip types. So the type of chipper used and how that impacts what the chips themselves look like. Okay, so I think we have time for one more question here. Did you measure dissolved phosphorus or dissolved DOC in the wood chip extractives as you were setting up your experiments?
We didn't measure phosphorus, but one of the things that we're up in the line for measurement is the total organic carbon. So we're still measuring that in order to, make a side-by-side comparison on the carbon concentration present on the wood chip, if that really influence faster removal or lesser removal. But yes for the carbon, not for the phosphorus.
All right. And so this project still has a couple more years ahead of it. So
Yes.
we hope to have you back with more
Yeah.
updates as we get more
Yeah.
through the microbiome and other aspects of this project. So we hope people stay in touch, stay tuned for more updates as we kind of fine tune this. We'll also have some upcoming outreach that'll focus on potentially using corn cobs within
Ah,
bioreactors. So we're trying to kind of explore what works best, how we can lower the cost of installation, and improve efficiencies of these systems. So thank you, Clarisse. This was
thank
absolutely
you.
fantastic. And thank you, everyone, for your great questions. Again, this is recorded, so feel free to go back and review any of the information Clarisse shared today. If you've got future questions, you can always reach out to her directly here at Iowa State. So thank
Yeah.
you so much, everyone, and have a great afternoon.